Hello guys, how you all doing? I hope you all are doing good. So last time we talked about dark field microscopy and before that we talked about bright field microscopy. This time we're gonna talk about phase contrast microscopy. So let's get started. But wait, before getting into phase contrast microscopy, let's see how the microscopes look like. See, like dark field microscopy, we are having laptop attached to it. Okay, but as you know, it is not always important to have a laptop attached to a microscope. We can also see through ocular lenses. These all are binocular lenses and through which we can see the image. So now let's get started with the principle of phase contrast microscopy. Now, as you know, when a cell consists of mitochondria, cytoplasm, and any other internal structure, there is always a difference in refractive index. Different refractive index, different cell density are already present. So what phase contrast microscopy is doing, this microscope is magnifying these refractive index. So when the images are formed using these variations, they are detected easily. They are clearly visible in the image that those refractive indexes are making the image more proper, because what we are doing using phase contrast microscopy, we are magnifying those refractive index. How we are doing that? It's there in the working of the phase contrast microscopy. Now see the parts of phase contrast microscopy. Apart from those basic parts, we have two major parts and one part which is just an accessory part which can help us to improve the image. Now see, there is a modified condenser and modified objective lens. If we compare this microscope to the dark field microscope, there we just have modified condenser, okay, which was allowing only a cone of light to pass through the image. But here we are having modified condenser as well as modified objective lens. Now modified condenser consists of annular stop and modify objective lens consists of a faceplate which is an optical disc. We'll be able to appreciate both of these modifications in the working of the microscope. But before that, let's see color filters also. Now see, whenever we are getting the image from phase contrast microscopy, we're always getting dark and bright images. Okay, so to improve the image, to get the colors into it, we can use color filters, but it is not necessary. If you want to use it, you can, just to improve the image. See now, before condenser, this is your annular stop and we use it before condenser. What is happening again? We are just allowing a cone of light to pass through the image. Like dark field, we are again stopping the amount of light. But what is the difference? The difference is, there we were almost stopping the amount of light that were coming from the center, right? If you have seen the images and have seen the slides in the dark field microscopy, we were stopping the amount of light that were coming from the center. But what is happening in the PCM, phase contrast microscopy, we are not stopping the amount of light that is coming from the center. Basically, it is the only part that we are allowing the cone of light to enter into the specimen. It's just the center part. Okay. And faceplate, which is placed before objective lens, is consist of an opaque disc and a phase ring. This yellow ring is a phase ring and apart from it is called phase plate, or you can say opaque disc. Now the importance of this phase ring will be decided in the working of phase contrast microscopy. Now what is working of my phase contrast microscopy? We have talked about phase working of microscopy from so many times. So let's finally get into it. So whenever this light source illuminates and a cone of light passes through condenser through annular stop, only a cone of light from the middle passes reaches the specimen. Now we know that cell already consists of different refractive indexes, right? So when light rays passes through the specimen, it has to get deviated. Variations in density and refractive index in the specimen. Why are these variations? These variations are due to cytoplasm, nucleoplasm, mitochondrial cell plasm, mitochondrial cell membrane, nuclear cell membrane. All these structures are having their own refractive index and their own variations. So what is happening? When light rays passes through these variations, they get retarded by 1/4. Why? Because these refractive indexes are higher, and so wavelength is getting retarded by 1/4. But when light rays is passing through the field, basically apart from cells, it is not getting deviated because there is no difference in the refractive index over there. So what is happening? When deviated light passes through the face plate and miss phase ring due to deviations. Now, when I'm saying that light rays are passing through cells, they are getting deviated, and light rays passing through field, they're getting undeviated. So now we have two type of light rays, which is first deviated light rays and second undeviated light rays. So deviated light rays, when passes through the face plate, they actually miss the phase ring, the middle part. Why? Because of the deviations. And those undeviated light rays coming from the background, coming from the field, they pass through the phase ring. And do you remember when I said that it is an optical disc? That means it has to do some with the optics of the light. So these undeviated light rays get advanced by 1/4. Now can you tell me the wavelength get retarded by 1/4, and there are also light rays that are get advanced by 1/4. So what is finally happening, guys? They are now half of wavelength out of phase because one is retarded by 1/4, one is get advanced by 1/4. So how much the difference is now? Isn't it half? Because one is getting retarded by 1/4 and other is getting advanced by 1/4. Why is it getting advanced? When it is passing through the phase ring, it is getting advanced because that's an optical disc. And when light rays are passing through the specimen, they're getting retarded by 1/4 of the wavelength. I hope that's clear. Now, it is not important that image is formed by the deviated light rays. It is, see, it's simple to predict that deviated light rays will be making the image, but it's not like that. So undeviated light rays when passes and undeviated and deviated light rays when passes through the objective lens, those undeviated light rays coming from the field makes the field of the image, makes the background of the image. Okay, so what is happening? It'll be bright. Now why it'll be bright? Because undeviated light rays when reaches the objective lens, it forms the background. But when undeviated light rays meets with the deviated light rays, they both cancel out each other and then they form the image. I hope that's clear. So when undeviated light rays are undeviatedly reaching the objective lens, they're forming the background and so it appears bright. But when undeviated light rays meets, like see, see here, undeviated light rays when meets with the deviated light rays, they both cancel out each other because they are half out of the phase and they form the image. So the image appears dark in color. And due to the different refractive indexes, different kind of internal structures are formed, and so a clear defined image is produced. I really hope that the working of phase contrast microscopy is pretty clear to you all, guys. If you have any doubt, show them in the comment section. Next time we'll be talking about some of the advantages, disadvantages, and the images formed by the phase contrast microscopy. But till then, bye bye, take care, and follow for more videos.